to go through. Which one of the following best describe nucleic acids? What's the nucleic acid? You know what's the nucleic acid? Talking about DNA and RNA. Which one describes the nucleic acids and nucleotides? Which one of the following best nucleic acids are monomers? What's the nucleic acid? You know what's the nucleic acid? What is a monomer? Building blocks. So nucleic acids are monomer. A monomer nucleotides. Nucleic acids are monomer of nucleotides are monomers of nucleic acids.
What if I can build an app that works my way?
They're saying that nucleic acids are monomers of nucleotides. No, nucleic acids are bigger than the nucleotides. Because when you have the nucleotide, yes, when you have the nucleotide, you form what we call the nucleic acid. You form DNA. Remember, you have phosphate, sugar, and nitrogenous base. So this is a nucleotide. So when you have many of them, you form what you call a nucleotide. It can either be DNA or RNA, depending on which sugar. So here they're saying that nucleic acids are monomers. Are monomer nucleotides? No. Nucleotides are nucleic acids, correct. But let's find out if there is a more correct answer. Nucleotides are large molecules and nucleic acids are small molecules. No, are small molecules because it is one. When they combine, they form large molecules. That is DNA and RNA, which we call the nucleic acids. You see? So this one also goes off. Then they're saying that nucleic acids and nucleotides are bases. No. So you see that the best correct answer is this. Nucleotides are monomers of nucleic acids, meaning that nucleotides, they make up the DNA and RNA. Do you understand?
Yes.
Did you get it right?
Yes. Yes.
Okay, they're saying that if 10% of bases in the molecule of DNA are adenine, what is the ratio of guanine? You see, we did this. Remember we said that if 10% of bases are adenine. So they are looking for adenine versus guanine. So it means that, remember, when you're doing base calculations, we say that A goes with T. If A is 10%, what is T? It's also going to be 10%. Because if you have 10 left legs, how many right do you have? 10. Do you understand? Because they are complementary. So it means that if I have 10, if I have 10 plus 10, it's going to give me. Remember, this is percentage, which means it's out of 100. So it means that it's going to be C goes with G. You know that. So it means that the balance of 20, the balance of from 100 is going to be for these two. Therefore, I'm going to say that 100 minus 20, which gives me 8. 8 is for these two. So since both of them are equal to each other, therefore I'm going to say divide by 2. So it's going to give me 40. So it means that this is going to be 40. Even this one is going to be 40. I see. But they are saying, what is the ratio of what? Of adenine to guanine. So I come back here, I look for adenine, which is 10. And then guanine, which is 40. Are you seeing it? And then now I have to look for the simplest ratio. So I'm going to divide by what? To get the simplest ratio, I'm going to divide by 10. Divide by 10. I'm going to get 1 here. I'm going to get how many here? 4. So do you have that ratio? What is the answer?
Yes.
D. So D becomes the most correct answer. I see. Yeah, did you get it right?
Yes, sir.
Yes, sir.
Okay. We go to the next question. Good morning. You got the next question. They're saying the section of DNA molecule that code for a specific characteristic. How do you call this section of DNA that code for a specific characteristic? Meaning that what are those sections which determine our characteristics in the body? What are those? Why do we resemble the way we resemble? And those sections, those parts are from the DNA. How do you call those sections? Nucleotides? Gene. Call them genes. Yes. So yes, correct. Genes, those are the sections of DNA that control a particular characteristic. Sometimes we say a particular trait. Who is that one making noise? Okay. When I'm gonna mute you if someone is making noise. Okay. Okay, you unmute when a question is being. The phase in the cell cycle that occurs before cell division. Interphase. Someone said interphase. Interphase. Are you getting it? Yes. The nitrogenous base is found in messenger RNA, not in DNA. People online, what's the answer? What's the answer? Unmute and tell us the answer. You said what's the answer? Uracil. Uracil. So you have to know these terminologies. It was just a simple test, just to show that so far now DNA is a little bit fine. Today we're supposed to do a second test. Supposed to do a second test after the other part of coverage. So we're gonna cover it, then we finish up, then we do the second test. Yes, you can do it home, but when we go through, we're able to go through and then get the what? The perfect answers. Let's go to guys. When it comes to this, you have to take note of this. When they say A only, B only, both A and B, none. Don't say A, don't say both, don't say B. You say A only, B only, both A and B, or none. Is it clear? Yes. They're saying car is genetic material. Which car is genetic material?
Chromosomes.
Chromosomes. They carry genetic material.
B.
What about genes? Genes are the genetic material we are talking about. So it means that chromosomes carry genes, basically. So bond that hold together in DNA molecule. Bases that hold nitrogenous bases together in DNA molecule is weak hydrogen bonds. Where do we find bonds? Find peptide bonds. Peptide bonds are between the amino acids, between two different amino acids. So when the bond between the two, we call them what? Peptide bond. Yes. Do you remember that? Do you remember that? Okay. So if you check here, if we check here, we see that, yeah, these are the bonds we are talking about. See, this is a polypeptide chain. And this is amino acid, this is amino acid, amino acid, amino acid. But these amino acids are joined together by a specific bond, which we call peptide bond. So I think now we hear that the peptide bond, you know where they are located. Discovered the structure, the helical structure of DNA. Who discovered the helical structure of DNA? Remember, there were 4 people who were involved in the discovery of DNA: James Watson and Francis Crick, Rosalind Franklin, and Maurice Wilkin. So here You see that this James Watson and Francis Crick. So both of them were involved. So basically, in this case, the actual answer is going to be both A and B. You understand? Yes. When you go back to your distinction material book, it's stipulated there very well and highlighted. You see it. Yeah. These are the people they're talking about. James Watson, Francis Crick. You see, they were working together so that they're able to discover the of the distinction. Sorry, the what the structure of DNA. You see that these two, they're also working together. But in this case, who described it, that double helix with the complementary base pair, is James Watson and Francis Crick. So if you come back here on the question, it is on page 21. If you come back here on the question, we see that Francis Crick and James Watson are too far. Yeah, you see that our answer, let me close this one. Our answer is what? Is James Watson and Francis Crick. We got the next question. We go to this one now. The diagram below shows or represents the part of nucleic acid or molecule, and answer basically the questions that follows. Identify the nucleic acid diagram above. What is this? DNA. Is it true? Let me hear from there. And what is being shown there? We say that is DNA molecule. DNA molecule. That. They're saying give one visible reason for your answer. Why do you say that this is DNA? Double-stranded. Yes, okay. Is there any others? With hydrogen bonds. Yeah, even even RNA has adenine, isn't it? Yeah. So it's double-stranded with weak hydrogen bonds. It's double-stranded hydrogen bond. Then we go to the next question. Then we got the next question. They're saying that did you join the class as you were coming? I thought that you were in the taxi. That's why I sent you a link. Oh. Then they're saying that label the following parts. What is part 1? What is part 1? Phosphate. Is a phosphate. Phosphate. What is part 2? Serious? Let me reload. Let me see. This one's no longer answering. What? Why no longer answering?
Deoxyribose.
Is deoxyribose?
I'm saying deoxyribose, I think.
Yes, it's deoxyribose sugar. Ne? Yeah, okay, it's deoxyribose sugar. Then that is part 2. They're saying that nitrogenous base 3. If this side I have adenine, what do I have? What is the collective name given to part 1, 1, 2, 3? Part 1, 2, and 3. Part 1, 2, and 3. How do we call? Basically, they're saying group. How do you say group in isiZulu?
Nucleotide.
Yeah? Nucleotide.
It's nucleotide.
Yes, it's a nucleotide. Okay. Okay. They're saying that they group all of them. The phosphate, the deoxyribose sugar, the nitrogenous base. How do you call it as a whole? You understand? Kaufera. All of them. You understand? How do you call it? What? Nucleotide. It's a nucleotide. nucleotide. And that's why I say that is the building block, see? You understand? So state two structures in non-dividing human cell where DNA is found. State two structures in a non-dividing human cell where DNA, they're saying human. Do you know what it means? Do you know what it means? They say human. They're saying animal cell. Animals. Yes, because the humans belong to animals. So you can't talk about chloroplastic DNA. So you can only talk about the nuclear DNA, the nuclear DNA and the mitochondrial DNA. But they are saying name two structures. They're not looking for the type. They're looking for where they are found. So it's going to be the nucleus and the mitochondria. So this is what I are talking about. If you go back our distinction material book, you find it here, saying that the type of DNA are based on. Oh, it's fine. You can just look here, unless you want to add something there. Yes, is nucleus, nuclear DNA, it's in the nucleus, then mitochondrial DNA, it is the mitochondria. So they are looking for the structures. So basically, it's the mitochondria and the nucleus. So this can't be found in a human cell, can't be found in the human cell. That's why it's out. Name one difference between nitrogenous bases found in DNA molecule and the RNA molecule. It's uracil. Uracil where? And then DNA. DNA is what? Thymine. So it means that DNA is thymine, while RNA is what? Uracil. Yes. Sorry. So it's done. You see that these questions, if you practice them, you get them right. Did you do them? Yeah. Yes. So if you practice them, you're gonna do them. Yes. Because you didn't do them. Yeah. However much you're going through them, we keep repeating questions. The more you repeat them, the more you get used to them. And when they bring them, you are able to answer them correctly. Yeah. But sometimes you might not get enough time to repeat them together due to the coverage you're supposed to cover. Okay, the diagram below shows DNA profile for 6 different 6 members of family. Okay, parents of Zintle, Ayanda, have 2 of the children are biological offsprings, while the other 2 children are adopted. What does it mean? It means that these kids, they are not what? They are not, those who are not biological. The profile is not the same. You understand? If you look at these profiles, you'll find out that the profile here and this must match with the kids. But since this is paternity test, you're looking for the parent, the father and the mother, you have to check the corresponding, I said it right? You have to check the bands which correspond for both children. All of them must be cleared. Okay, let's go. Let's start with Lindy way. This with this, isn't it? Yes. This with this, isn't it? Yes. This with this. This goes with this. This comes from here. This comes from here. You see that all of them for this child, they are covered. If there is one which is not covered, then it means that is just a relative. It's not a father. They're not father and mother. So that's why you say that if you subtract the mother, the remaining bands of the father, the remaining bands of the child must correspond with the bands of the father. Or if you subtract that one of the father, the remaining bands of the child must correspond with that of the what? The mother. Yes. Okay. So it means that Lindy is a biological child. Let's go to Google. Nope. 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 This one maybe correspond with that. I don't know. Nope. 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 Nothing. It's only one. So the remaining bands, yes, this one, if I take the father, the mother, the remaining bands of the child does not correspond with the father. So this one is adopted. Then we go to this one. This goes with this. 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 This goes with that. Isn't it? So Bandile is also a child. Then this one, you see that, nope, nope, yes, goes with that, nope, nope, nope. You see that, okay, this one goes with that, but this one, nothing. You see that even if there are two, but still all of them don't correspond. So it means that Lindiwe and Bandile are the biological children of the parents. And then Gugu, and then Letswe. Please, Letswe. Lizwe.
Yeah.
Oh, Lizwe. Yeah, they are not, they're just adopted. Yes. As they're saying, which children are biological offspring of Zintle and Ayanda? We say that it is Lindiwe and you see how we get it. Then you're saying, give the explanation for your answer using the evidence from the DNA profile. You see? So you tell us that the DNA, the bands of Lindiwe and the Bandile, yes, correspond with Zintle and Ayanda. Therefore, Lindiwe and Bandile are biological children. Yes. Apart from patented test, state to any other way in which profile is being used in human beings. They are basically looking for the uses of DNA profiling apart from patented test. Identifying crime. In the case of fire, what? Forensics. For identification of human remains. Talking about personal identification. Yes. They're saying that questions below are based on DNA profiling. What is DNA profiling? Because this one is no longer being asked. But we're no longer asking you, Steph, what's DNA profiling? You just need to know what does it do. Basically, is a technique which is used to identify someone's unknown DNA profile. Give evidence. DNA evidence of a murder suspect was found at the scene. Give two possible reasons why the suspect might not be found guilty in court by referring to DNA evidence. Now, this question is just an application question. Oh, you just think about it. Basically, they're looking for why DNA profiling is not 100% reliable. Because they can ask you, why is it reliable? Is it conclusive? Yes, it can be conclusive. Conclusive means that, can I use DNA profiling and I conclude that you are the person who did this, where now you are the son of this, after using DNA profile? When they do DNA test, do people confirm that these are our parents? Do they do that? Yes. But I can say no. I can say no. So what are some of the reasons you wanna base on to say no, that the results you have given me, they are not correct? What's the answer?
Because they might be contaminated.
Contaminated.
In the during collection.
Contaminated with DNA. Ah. Any other?
I think DNA can be just with the
Okay.
Like I can transfer it.
Okay, since this is a suspect, this is a suspect, and they collect this at the crime scene. What if this person was framed? This individual can be framed. That's number one. Number two, there could be human errors. You have analyzed it wrongly. Number three, the hospital or whatever used might not have all the necessary equipment. It could be human error. Yeah, hospital might not have necessary equipment to carry out DNA profiling, but you have to modify it to suit the question. And then it can also be false evidence, meaning that the person can be framed. You understand? Yes. The person can be framed, and then they say that, you were there, yet someone took maybe your blood or your saliva or your anything which has your hair or what to the crime scene, because they want to frame you up. So yes, it might not be conclusive because of human errors. It can be someone can frame you. Maybe they are not enough to carry out this process properly. Yeah, that's what they're saying. All right, I think you have this question in your book, in your distinction material book, isn't it? It's there. It's page 8. Okay, let's find out what they say. Identify the following molecule W. What is molecule W? What is molecule W? Oh, how do you call this molecule W? How do you call this molecule W?
DNA nucleotide.
DNA nucleotide. Remember, it's a phosphate, a sugar, and nitrogenous base. So it becomes, molecule W becomes a nucleotide. Then number 2, they are saying that nitrogenous base V. Nitrogenous base V. If this is this, ne? And this is thymine. This thymine came from here. So what is V? Do you get it? Do you get it? Sis, Vosile. Vosile. Do you get it? Okay. And then they are saying that nitrogenous base V, which process is shown in the diagram above? Translation. Serious. DNA replication. Correct. And in which cell does this process takes place? Where in the cell does this process takes place? In the nucleus. When does this process occur?
During interphase.
Interphase. During interphase. Those questions are there. Those questions in our definition material book. So you guys make sure that you read this book, because there is no way you're gonna fail. There is no fail. You see, what is it? When does it occur? During interphase. Where does it take place in the nucleus? Those questions are there. So there's no excuse to give me that you have failed. There's no. Describe the process mentioned above. So it's the same question here. They are saying How does it occur? Oh, how does it occur? They're saying that describe the process. That's why you see 6 marks. DNA double helix unwind, weak hydrogen bonds break to form two separate strands, each acting as a template for formation of new strand using free floating DNA nucleotide from the nucleoplasm, whereby adenine goes with thymine, guanine goes with cytosine. 2 new DNA are formed, each with original strand and 1 new strand. The two new DNA are genetically identical. This process is controlled by enzyme. So this process, we have repeated it. You have to memorize. There's nothing special I'm going to say or how I'm going to help. This process, things related to process, ne? It's not analysis or what. Things related to process, to describe a process, it's you to memorize it. Because there is no paper which is going to come without a process. There are around these 6 processes which we bring. At least you're going to bring 1. In most cases, we bring 1 or 2. In most cases, bring 2, not 1. So it means that at least you go to the paper when you know that I have 12 marks. Those 12 marks, I have them. So you have to be keen. Yes. Aha. Then the next, so let's that you memorize.
Yes.
Zelda?
Yes.
Yeah, even if you have a lot of problems, sometimes you put the problems aside, forget about the problems, focus on your work, because if you don't pass it, the problems are going to come. Let's continue. Explain the biological importance of this process. Why is DNA replication biologically important? So what? It's true, but so what? To double the genetic material, so what? So what is the importance of DNA doubling its genetic material? It doubles the genetic material so that, remember in mitosis, if a cell is 22 chromosome, the cell must divide to form 22. What is this? What does it mean? It means that it has doubled. So it doubled before, meaning that now this cell, the normal cell, is supposed to be 22. So before it divides, it doubles. Then it becomes 44. You see? When it becomes 44, now this one becomes 22, this one becomes 22. That's the normal state. If this one wants to reproduce, to divide again, what does it do? It does DNA replication. So once it does DNA replication, so that now it increases to 44, so that when it divides, this one is going to be 22, this one is going to be 22. Do you see that the number is kept on maintained? Do you see it?
Yes.
So basically, we have DNA replication so that the genetic material is doubled. When the cell division occurs, can maintain its number of chromosomes. You understand? Yes, each cell can maintain the number of chromosomes. Why should it double? It doubles so that each new cell can have the same number of chromosomes. Yes. Tabulate the difference between DNA and RNA.
Ribose sugar. Sugar.
Ribose sugar.
Yes. DNA is double-stranded form.
Double-stranded.
And RNA is single-stranded.
RNA is single-stranded.
DNA contains ribose sugar.
We are done with it.
RNA contains ribose sugar.
That one we are done with it.
DNA contains
DNA has thymine.
RNA is usually.
Yes. People online, I don't hear you. You are busy quiet as if we are listening to the service that the pastor is preaching. This is not a service, Baba. Supposed to engage. All right. So, guys, that was the paper. I think the paper was simple. I don't see any challenge here. Everything which was here, we had covered it and we had gone through it via our distinction material book. And I think